Hello, everyone, and welcome to today's RettEd webcast. This is Paige Nuez, Director of Family Empowerment at rettsyndrome.org. Our board of directors and all of us at rettsyndrome.org want to thank you for being with us today and for your support and engagement. We know that continuous awareness and empowerment is critical for our rare disease, and your interest in our research programs and pushing the limits of science is a refreshing topic, I think, to show the continuity of research during these unusual times. So we had booked Dr. Muotri to be with us a number of months ago, and I am really grateful that in this time of great disruption, that he stayed true to you, our community, and to our foundation to be with us today to talk about the continuity of his work and that of his team at his lab in San Diego. So there we go. Okay, next slide. Yeah, so in a few minutes, we're going to talk with Dr. Muotri, and he's going to share with us about his work in Rett syndrome, some of the incredible work he's doing with space labs, and answer any questions that you have for him today. I wanted to give a few updates before we get going. Before we lose everyone at the end, I wanted to remind our families who are in attendance with us today that we will come together again on May 26th for a Facebook Live session talking about ideas for modified aquatherapy at home. It's a little bit more closer to our home and a specific topic about things that you can do with your child as we come into the summer months in the Northern Hemisphere, and many of us are still spending a lot of time at home. So I hope you'll enjoy that upcoming event. I also wanted to share that we accelerated production of the communication guidelines, and that was a multi-year research project conducted globally to see how our kids communicate and what are some of the best practices worldwide, from low tech to mid tech to high tech. And this wonderful guideline is now published and available on our website. You can purchase it. Printed versions are a little bit slower to get shipped these days, but you can also access a digital version, and that is especially good for our families outside of the United States, where shipping can be very expensive. Okay, and we are ever grateful for the continuing support of our community to help our foundation continue to advance research, and especially to support families during this time with a lot of resources that we are producing thanks to our expert community to keep our kids engaged, communicating, moving, doing all kinds of things while at home without usual supports. Although we know that those are becoming available more and more, it's just not in the way that we're accustomed to. So we appreciate your support so that we can continue those programs, and we hope that you will consider participating in our virtual 5K. All you need to do is start a personal page on rettsyndrome.org, start fundraising for the Rett Racer virtual 5K, and as soon as you hit $100 raised, we will send you a Rett Racer t-shirt and hope that you will join us on June 6th. Again, you can visit our website to get more information about that. Okay, so how the webinar will work today, if this is your first time joining us, the audio is open to Dr. Muotri only, so you don't have to worry about background noise interfering with the presentation. If you look to the upper right corner of your screen, you'll see a control panel window, and we invite you to type all of your questions and comments into that field, and Dr. Muotri will answer your questions at the end of his presentation. And we do have the questions that were pre-submitted at the time of your registration, and he'll address those questions at that time as well. If we don't get to your question today, we will work with Dr. Muotri to get all of the writing questions answered, and we'll send out a QA document to those who are registered today. If the control panel window is distracting to you, you can minimize or move the window, and then you can expand it if you want to submit a question. Next point, the session is being recorded, and we will post a link of the recording to the RettEd webpage so that you can listen again or share it with others who weren't able to join us today. So don't worry, you don't have to spend your time taking notes. Just sit back, relax, and let's hear some information from Dr. Muotri, who I'm going to go ahead and change the screen over now, if I may. Dr. Muotri is the director of the stem cell program at the University of California, San Diego. He is on our scientific advisory board. He is a funded researcher with rettsyndrome.org, and his work in the field of neuroscience is extensive. He is received many, many awards in his field. And Dr. Muotri, we're seeing your screen, so if you want to go into slideshow view, that would be great.
Super. Thank you so much, Paige. And thank you for the introduction. Always a pleasure to talk to parents and families. Before I show my slides, just to mention in my bio that I'm also a father, and this is Ivan. This is my wife with Ivan. Ivan has a mutation in a gene called SETD5. It's also a chromatin-related gene, so it's kind of a similar family of genes that controls how the DNA is organized. So Ivan has a severe type of autism and epilepsy. So I kind of share my duties as a father and as a scientist. So I kind of understand all the struggle that the families goes through. And I feel that it is something that I can offer by explaining science in a very easy terms for the family so they can understand and follow. So I decided to talk about some of the previous research that I was involved regarding Rett syndrome, as well as some of the very exciting things that are cooking in the lab, just as a teaser for what is coming in the next few years. And we are very excited about that. So I hope you enjoy as well. So first, let me tell you a little bit about myself. I'm a geneticist. I actually got my PhD in human genetics back in Brazil, in São Paulo. And then I moved to California and I became a neuroscientist and focused on stem cell biology. The reason why I moved to California was to establish a human model. I want to understand how the human brain is formed. And that's a very difficult question to answer. Because the human brain is actually formed inside the uterus. So this is a picture of a pregnant woman and holding a very low resolution picture of the fetus, and a couple of pixels in there is the human brain. So we cannot see how many neurons are there. We cannot see if these neurons are active. We cannot see how the brain is formed, how the cells migrate. So if we cannot really recreate that, we don't fully understand. And I use stem cells as a model to recreate these very early stages of human neurodevelopment because they are so important in neurogenetic conditions like Rett syndrome, because that's exactly when the defects or the problems in brain anatomy and structure and organization and networks started. So if we don't really understand how everything started, it's hard to fix. And most of the biomedical research is actually focusing on animal models. And I was really frustrated with the animal models, especially for the poor translatability. This means that we can mimic or recapitulate some of the diseases, the genetic diseases, using these models. And sometimes, I mean, the models are good models, sometimes they're not so good. But one thing that we researchers learn is that especially for pharmacological treatments, you can cure animals with all kind of genetic defects that are similar to humans. But when you try to use those same treatments in humans, there is a 95% failure. So which means that the mouse brain does not fully recapitulate the human brain. Seems obvious, and it's so funny when I talk to families, they said, of course not. Why are you guys studying the mouse brain? Well, that's exactly the point. I mean, models are really important for science, and we didn't have any other models to actually study human diseases. That's why we rely on animal models. But gladly, stem cells, human stem cells, came as a new powerful tool where we can model some of these conditions. And so I started my lab here at UC San Diego in 2008, 2009, and that was when a stem cell method called cellular reprogramming were really picking up. So cellular reprogramming is a way to convert any somatic cells, any cell from your body, back to an embryonic stage. And you can do that by activating 4 genes. Sounds complicated, but it's not. It's actually very simple. These days, we have high school students doing that for us here in the lab. So it's that simple. And the guy who actually discovered that is a colleague, Shinya Yamanaka, and just 5 years later, he got the Nobel Prize because it's such a seminal discovery. So what does it mean? Cellular reprogram means that I can take a cell from someone with Rett syndrome and go back to their embryonic stage and recapitulate, from that stage, I can recapitulate their neurodevelopment. So I can mimic exactly the stages where when the brain is formed in a controlled condition in the lab. Of course, it's not perfect. As any model, it's a very simplistic representation of the reality, but we are talking about human cells, something that we never had before. And so when we start modeling Rett syndrome, we were generating all those brain cells, and we were looking for differences. And the question we had as a lab, what kind of differences should we expect in Rett syndrome cells? So fortunately, there was a couple of papers studying post-mortem tissues. These are brains donated by the families from their deceased family members, and we can stain those neurons and look for their morphology. These cells are no longer alive, so we cannot study electrophysiology or the behavior of those cells in vivo. But in a post-mortem tissue, you can do something that we call Golgi staining, which is just labeling some of those neurons, and we can compare the morphology. So on the top row here, what you have is neurons derived from a boy with Rett syndrome, and on the bottom, it's an equivalent in age control neurons from a healthy individual who died by something else. And I think it's very clear the complexity of those neurons. So neuronal cells, they are fully arborized. They have all these processes and spines. These are how they connect to each other to form circuitries, and circuitries generate behavior. So by looking at the individual cells, you can tell that the ones derived from Rett syndrome, or neurons carrying NCP2 mutations, they are not as arborized or as complex. You can even look at the soma size, the cell body of these neurons, and see that they are slightly smaller compared to controls. So something is not right here. I mean, they are just not on their developmental stage. They seem to be very mature compared to what they are supposed to be. So then, I mean, we isolate cells from a couple of Rett syndrome patients. We use the cellular reprogramming to push them back to this embryonic stage. And from then, we made neurons from those people. So those neurons actually show the same thing. I mean, they were very immature. This is just one of the analyses that we did. I think I'm not sure if you can see my cursor here, but anyways, the picture, it's a high magnification of a process. Y type means healthy control. RTT means one of the Rett syndrome subjects. So these are actually live neurons that we could recreate in the lab. And the number of processes or spines is very reduced in Rett syndrome patients compared to controls. That's exactly what we saw in the postmortem brain tissue. So our model seems to be recapitulating at least one of those conditions. So I told you that those spines and processes are what the neurons use to communicate to each other. And there is one way that we can visualize the number of connections that they make. And this is by using a technique that make the synapses or the connections between those neurons to fluoresce. So I have a video here where the neurons, the process of the neurons are in green, and the synaptic connections, we were labeling them in red. So you can actually see the puncta, and you can quantify the number of puncta that you see in the control neurons compared to the Rett syndrome neurons. And it's very clear that Rett syndrome neurons do not make as many synapses as we see in a healthy individual. So that was a very dramatic effect, and it's very reproducible. We see that in most of the patients with MCP2 mutations. We can actually quantify. As I said, we just count the number of red dots that we see per neuron, and we see that the number of synapses or connections are about 50% lower in rats compared to controls. So similar to the morphology, the picture that we have, it seems like Rett syndrome neurons are just immature compared to same age control neurons. But there is one observation that we did, and this was back in 2010, that I think was very exciting to me at the time. We were wondering if those differences are permanent. So if you wait longer enough, will they get away or not? And so we did this experiment. We wait long enough, and while the control cells start to make more and more synapses, the Rett syndrome neurons were stuck on that developmental stage. But if we challenge those with pharmacological treatments, and we use, for example, IGF-1, insulin growth factor 1, it's a molecule that's in clinical trials now, you can see that the number of synapses go back to normal. So that was very exciting because it points to the fact that some of those cellular defects, at least the ones that we are analyzing, they are not permanent, but can be reversible. So that's a very new concept in neuroscience. In the past, we were thinking that defects that happens during neurodevelopment cannot be fixed, cannot be reverted. But this data, together with other mouse data coming from Adrian Bird's lab, suggest that no, you can actually revert those defects if you know how. So you might say, well, why don't we are all taking IGF-1 as a treatment for Rett syndrome? So there's a catch here. When I add drugs in those neurons in culture, they don't have what we call the blood-brain barrier. This is a membrane that protects the brain from external molecules or pathogens such as virus. So the brain is really protected. And molecules like IGF-1 is quite big. So in vivo, when you take IGF-1, very few molecules will pass to the blood-brain barrier and reach out the brain. So we have to work with small molecules. And there are some efforts on trying to decrease the size of IGF-1 so it could penetrate better in the brain. So that might be one thing to keep in mind when I'm using this very simplistic model here in the lab. All right. So, but that's really encouraging. We know we can use this model to at least test some therapeutic ideas. And so over the years, my lab has evolved in this technology to create what we call brain organoids. And I'm placing this image here just to provoke you, because you're probably going to read in the media or in many places a mini brain in a dish. And this is probably the image that most of non-scientists will have when they hear something like as a mini brain. But the truth is that what we have is nothing comparing to that. That's why we prefer to call them brain organoids. It's a Latin word that means a smaller version of the tissue. So we actually have a very tiny piece of the brain that we can create from these stem cells derived directly from the patients. And as any other model, they do have their own limitations. I'm listing here just a few of these limitations. They are immature, they're not vascularized. The human brain contains 86 billions of cells. We can get about 2 million of cells in these organoids. We don't even know if we are growing them in the right culture condition. And similar to the animal models, we don't know how translational they are. If we learn something from these brain organoids, can we apply it to the human brain? We just don't know. But the science is moving quite fast in this area. There's many people excited with this technology. Again, it's a new human model, and the possibility to model genetic diseases such as Rett is very attractive. So there are more labs working on this technology than they were 5-10 years ago, which is really nice to see. And And since the science is moving fast, I mean, my lab is also been optimizing several of these conditions, and we actually could recreate some brain organoids that are functional, meaning that those neurons now can form very nice anatomical structures. For example, you see in this picture here that we see a cortical plate, something that we never get before in previous protocols. But most importantly, those neurons are forming connections. And those connections are forming small circuitries. And last year, we were able to measure these circuitries, and we figured out that they match human development, at least in a very superficial gross model. Of course, it's not identical, but they seem to feature some of the same things that we can record from EEGs. And we continue to push this model to make them more and more similar to the human brain. And one way to do that is really to accelerate aging. And one idea that we had is to send some of these organoids to space. I think that's why Paige was very excited about brain organoids in space. So under microgravity, these human cells, they suffer what we call accelerated or speed aging. So it might be that by growing them in space and bring them back to Earth, we could improve this model, make this model be more similar to a little bit older human brain. So that's one of the reasons why we are now creating these brain organoids at the space station. So, but they are also good to screen other candidate drugs. They're actually a more rigorous model than we had before. And this is something that we have now here in the lab. We create a drug screening platform using this brain organoids so we can test many drugs. And I'll just summarize some of the recent data that we have and why we're excited. So what you have in the graphic in your left, it is in blue, the number of synaptic connections made by a healthy control organoid. In red, you have one of these Rett mutant organoids. You can see the reduction in the number of synapses, almost about half. And then a series of drugs. And the last one is IGF-1, a series of drugs that we are using to see if they can improve the number of synapses. And all the ones that have a star on top are the ones that, after multiple trials, could revert the number of synapses back to control levels. So this is very exciting because give us other therapeutic perspectives, but also points, because we know what these drugs are doing, points about what kind of defects are happening or neurotransmitters that are defects in the Rett syndrome brain. So we are learning more about the Rett syndrome brain by performing these experiments. And we selected two of the best drugs. This is the graphic here on the right. One drug is nifuracetam. The other drug we call PHA. These are compound tests, compound drugs, sorry. And those two drugs, we challenged them with very rigorous experiments to see if they can actually restore networks in Rett syndrome. And this is just one piece of the data showing that these two drugs can actually fully revert back all the synaptic as well as the network defects that we see in Rett syndrome. So why this is good news? Because we now think that different from the past models that we had, that this model really recapitulate or give us a little bit more of a confidence that these drugs are closer to the human brain. Many of the previous drugs, if you start here in your left, could not restore all the way to the brain organoids, but those two could. So we're now looking for a potential way to do like a small clinical trial to test those drugs in vivo. At least for nifuracetam, we know that it's safe in humans, and it's a drug that's free of IP. So that's good news. The bad news is that since it's free from IP, there is no pharma interest on testing these drugs. So we really have to rely on investors and families to raise support for those clinical trials. So that's what we are looking for. And we can go back on how we are planning to test those drugs later on. So the other thing that these brain organoids are really good, it is to test therapeutic opportunities or interventions that are non-pharmacological, for example, gene therapy. This is the work that has been sponsored by the Rett Syndrome Research Trust. And I was part of this consortium that were analyzing the efficacy of gene therapy ideas. And I'll share some of the data that we have, which I think it's very interesting data. We use these adeno-associated vectors. These are viral vectors that carries the NCP2, the correct form of the NCP2, to deliver the gene back into the deficient neurons. So the value here is that we are testing this in human neurons, in human Rett syndrome neurons. So it's not in an animal model, it's not in a monkey, it is in actually human neurons. So the pictures here are showing some of the neuronal process in red. And again, the connections or the synapses that those neurons are made are in white. And when I label them as a 2M, this means that these cells are coming from an NCP2 duplication case. So they have twice as many NCP2 proteins in there. So they make more synapses than normal. So the control are here and the rats are there. So we actually see a very nice amount of synapses when we compare a healthy individual. We have lower number of synapses in rat, and we have a higher number of synapses in an NCP2 duplication case. So as a control, we know exactly where the number of synapses should be, and we don't want to have more synapses, and we don't want to have a lower number of synapses. So this is one of the data from one of these adeno-associated vectors. You can see that the number of puncta, or the white dots in those images, increase compared to red. And you can quantify here, and you can see there is a partial rescue. Why do I see a partial? We see that it's elevated compared to the Rett mutant, but it doesn't reach the same level as the controls. So why this is happening? In these experiments, that's the part that I think it's more fascinating. In these experiments, the number of neurons that were infected with the adeno-associated vector is only 1%. So it's 1% of the neurons receive the vector, and we already see an increase in the number of synapses. So to me, this is very encouraging. And we move on and test those in brain organoids. So we created these brain organoids, we plate them on dishes that contains printed electrodes in the bottom of those dishes, so we can actually see the activity of these neurons as we infect them with the viral vectors. The viral vectors infection also have the EGFP, so this is the green fluorescent marker, so you can see the neurons that are labeled, they will fluoresce in green. So you see that it's only a spot there in a brain organoid that has been infected, so it's not the entire brain organoid. And it's enough to generate some activity. We have these activity maps. Every dot in there is one of the electrodes. And when we quantify, we see that by increasing the amount of the viral, you increase the number of functional neurons inside the brain organoids. Again, I mean, we are talking about less than 1% of the neurons that are infected, and we already see a rescue. This is great news for gene therapy. If we are able to put these vectors into a brain of someone with Rett syndrome, it probably means that we don't need to infect 100% of neurons to see a positive impact. Maybe just only a fraction of neurons that receive the gene therapy vector would be enough to provide a positive outcome for patients with Rett syndrome. That's why my lab and other labs are very excited about this possibility of a gene therapy for Rett syndrome. So you can actually rescue some of this connectivity. So the final idea that I want to share with you guys, and I think this is really, really promising, it is this idea that neurons are defective, but neurons are not the only cell types in the brain. So in this cartoon here, I like it because I'm illustrating different cell types that we see in the brain. Your neurons are there. You can see the oligodendrocytes in blue. These are cells that help to myelinize the neurons. And you have here in green your astrocytes. The astrocytes is part of the glia cells. Glia cells means from Latin as glue, so they keep the brain tied together. And in the beginning, we had the impression that glia cells are only supportive cells. They're just there to glue the neurons together and nothing happens. But more recently, we know that they are actually very active players in helping neurons to form connections. They are actually very close to the blood vessels. So when you take a drug that goes to the brains, the astrocytes are the first ones to sense those drugs even before the neurons. And they can communicate with neurons to tell if the environment is a good environment. So astrocytes are quite important. And a couple of years ago, the lab from Gail Mandel made the seminal discovery. They showed that glia cells, especially astrocytes, are very important for Rett syndrome. If you remove the MCP2 or create a mutation of the MCP2 only in astrocytes, you can recapitulate many of the features of Rett syndrome. So my lab was able to confirm that using our human model. What I'm showing here is going to be when we co-culture astrocytes with neurons. And if you co-culture, the neurons are here in green and the astrocytes are in the background. You cannot actually see the astrocytes. I keep them without a color so you can appreciate the neuronal morphology better. So these are neurons that are surviving on top of healthy astrocytes, mimicking exactly what happens in a normal brain. So you can see that those neurons are arborized, they have lots of processes, they are making connections with other neurons. So now, if I move those neurons and I plate them on Rett syndrome astrocytes, that arborization goes away, so the neurons look kind of bipolar, very mature looking neurons. The same happens if I plate Rett syndrome neurons on top of Rett syndrome astrocytes. We don't see that they mature and evolve. They actually look like very mature as well. But here is the big surprise. Now what we are doing is we are moving the Rett syndrome neurons and place them together with normal, healthy astrocytes. And you see that the arborization comes back again. So remember, we can no longer now distinguish between Rett syndrome neurons and healthy neurons. The only distinction between those two is because one of them carries mutations in the MCP2 gene. But at the morphological level, and we also have data on the physiology of these neurons and number of synapses, they look absolutely normal, which is amazing. I think I like to say that it's a punch on the genetic determinism. So you might have the mutation, but if you are in the right environment, those neurons will thrive and form the synapses and connections as a normal health neuron would do. So what is so important about these astrocytes? How come these astrocytes can actually revert those defects in Rett syndrome? We're still investigating. We know several of the molecules that might be helping on making this Rett syndrome back to a healthy state. We are also investigating lots of neurotoxic factors that Rett syndrome astrocytes release in the media that might affect neurons. So this is another therapeutic opportunity. If you can block these neurotoxic factors from the mutant astrocytes, maybe you can help the Rett syndrome brain. But perhaps one of the things that can be done, it is this idea of cell transplantation. And this is quite new for Rett syndrome. I bet many parents have never heard about it, and it's because nobody tried it before. So the idea here, or the concept that we're working with, it is to create health astrocytes, and this can be done using cells from the same patients that we correct the mutation, or from a donor, and we expand those health astrocytes, and we transplant back the health astrocytes into the Rett syndrome brain. So the model I'm showing here is a model using these brain organoids. So of course, I mean, we need to run a lot of preclinical data to establish that this is secure enough before we move into a cell transplantation in humans. But that's where we are right now. So those balls or spheres that you see, these are spheres derived from a normal individual. On your left, you have brain organoids from a control individual, and in your right, you have from a Rett syndrome mutant individual. And you can see that the Rett syndrome one is smaller compared to the controls. This is again, I mean, we see this all the time. The Rett syndrome brain organoids are always smaller in diameter compared to controls. Sorry. And so we label these astrocytes in green. We are using the EGFP, the green fluorescent protein, to visualize these astrocytes, and we transplant them back into these brain organoids, and we wait. Right now, we have a 30 days post-transplantation, but we have other brain organoids in culture going on for longer. And the good news is that not only these astrocytes can be transplanted back to these organoids, but they also spread out through the organoids. So they are not totally limited to the site of injection. They have an ability to migrate out from the site of injection and really repopulate these organoids. So what they do when they are inside these organoids. I'll show you one data that I think is pretty cool. These are pictures of, again, human astrocytes derived from a normal individual that we transplanted back into a Rett syndrome brain organoid. And when we look into the neurons that are close to the transplanted astrocytes and we measure their size, we see that the fact that they are nearby or close to the healthy astrocytes makes them at the same size as the control neurons. Remember, I told you that those Rett syndrome neurons are smaller. So that's not only we can recapitulate that with the organoids, but we can rescue that by transplanting a normal healthy organoids back to these normal healthy astrocytes back to these Rett syndrome organoid, and we can see a rescue on their size. We are continues to perform other experiments. We are even trying in vivo to transplant these organoids into the mouse brain to see if the human cells can actually restore some of the defects observed in the mouse model as well. So if all goes well, I mean, this might be a very nice opportunity for Rett syndrome. Initially, I was very hesitant to take this as a cell therapy for Rett because I'm not a neurologist, so I was always worried about opening brains and doing injections, things like that. But when I talk to my neurosurgeons colleagues, they are always very positive, saying that no, they do this all the time. It's a very safe procedure, and especially if the astrocyte stays there for longer periods of time and migrate throughout the brain, this might be a single treatment for the rest of the life, which is really cool. So this kind of strategy is very high risk. Nobody has done that before. And again, I mean, we have some preliminary data pointing to some of the efficacy in our models. But we really need to push this forward. And I'll send a message to the families in California. Most of these studies was actually supported by CIRM, California Institute for Regenerative Medicine. There is going to be a ballot to renew. This is a Proposition 71 in November. Please vote yes, because this is the organization that actually fund this kind of work. And this is how we got this preliminary data. Otherwise, it would be impossible to perform these experiments. Traditional funding agencies such as NIH are typically averse to these high-risk strategies. So CIRM is the way to go. If we got a CIRM renew, we're going to have $5.2 billion, and $1.5 billion should be dedicated only for neurological conditions. And I think we are in a good position to pitch for this organization to continue to support this therapeutic idea. So I really think that we should support CIRM and hopefully would be renewed so we can continue this work. I think that's all I have to share with you guys. I'll be more than happy to answer some of the questions. Paige, I don't know if you want to take it from here, or we can read together the questions and I can answer them. Let me know.
Okay, thank you so much, Dr. Muotri. That is incredible and very refreshing to see that your work is continuing in these times, and really nice to get back to some of the fundamentals of what we're trying to achieve in the field of Rett syndrome. We've been very much focused on taking care of our families right now, and you are taking care of our families in such a different way, and we're so impressed with your work. It is pretty incredible.
Thanks, Paige. Thank you.
And thanks for those slides. And if anyone has any questions for Dr. Muotri, please type them into the question field now. Organoids, very new concept for most of us. And if you had any questions, if you want some further clarification, now is your chance to ask. I think we had 2 questions that came in, Dr. Muotri. I think you already have those on hand. Would you like to address those while the audience has a minute to submit some further ones?
Yeah, the questions that came are all related to the organoid model, how valid they are. So as I alluded, they are recapitulating these very early stages of neurodevelopment where it's hard to have in other models. For example, a mouse brain would be fully formed in 20 days. The human brain really takes 6-month gestation to be really formed. So this is low pace of the human brain. It's hard to recapitulate in a mouse model. So that's, I think, the value of these brain organoids. We can take our time to really see how the cells migrate, mature, form networks without skipping any developmental stage. And by learning how the human cells do it, we're going to learn when defects appear. So again, comparing Rett syndrome organoids to healthy control organoids allow us to see when those differences appear, what are those differences, can we do something about it, and hopefully in the future we'll have some insights about how those early stages differences will lead to alterations in cognition, alterations in behavior. So that's I think the value of this model.
Great. Thank you. It's a lot of discovery work. There's a lot of discovery work that needs to happen before things can come into play for our children. And we appreciate, again, the creativity and you thinking of all the different avenues to explore in the brain. It's incredibly complex. And the fact that you're looking at all of the different pressure points of where you can test in Petri dishes, in animals, IPS lines, stem cell. It's incredibly comprehensive. And we know it takes time and it takes support.
Yeah, I'll be more than happy to. I know some of the families will digest that, and the presentation will be available online. So happy to follow up on questions and clarifications. I think some of these concepts are quite new and will take some time to digest.
They are. I think, especially about organoids. This is a very new concept, and I'm not sure that everybody really got their head wrapped around what those are. And I encourage everybody to listen to the presentation again, and please submit your questions to me. This will be an ongoing conversation. As you know, Dr. Muotri and his team, incredible team, who are still working under these circumstances to the extent that they can, are always happy to hear from families. And that's what we appreciate about you so much, is that all of your work is always focused on our children and how your work will translate to a brighter future for them. We really appreciate you sharing your personal story as well. I think that we hope that your work benefits all of our children who suffer with.
Fingers crossed.
Okay, so we do have a couple questions that have come in while we're finishing up, and one of our parents is asking if you are able to share at all about the 2 drugs that you talked about and a little bit more information about those. Is that something that you're able to share with us?
So I think these are under review now. It's going to be published, and then all the data would be available. I was a little bit hesitating to even mention these drugs because I know how the families get super excited about them. But especially because these are compounds that nobody's actually producing them right now. At least nobody that I trust. So I don't want the families to run to the pharmacy or try to go to the black market and ask somebody to produce them or get them. So please don't do that. Those needs to be validated first in a clinical setting. But these are drugs. In a summary, these are drugs that modulate neurotransmitters. Those 2 drugs are not specific, so they are acting in several receptors in the brain, several neurotransmitters, but they were able to normalize most of the alterations seen in the Rett syndrome brain organoids, including the network formation, which I think is really cool. So one of them, nifuracetam, has been in several clinical trials for other diseases and never passed phase 2. Which means that they fail to support whatever condition they were designed for. It doesn't mean that it won't work for rats. But the good news is that the fact that they are already passed up to phase 2 and 3 means that they have been tested in humans for potential toxic effects, and they are pretty much safe. That's what I like about it. Of course, it's a dose effect. If you take too much, might show some side effects as any other thing. But we are excited. We are planning a small clinical trial to see efficacy. We believe that the FDA might let us run already with a phase 2 because of the toxicology for all the studies has been completed. So that's where we are right now.
Okay, great, thank you. And some of the other questions were related to timeline for trials, and in addition to the other.
Yeah, so it is all really dependent on. So we of course, everything is slowing down because of the COVID. Even our clinical collaborators are now so busy with this COVID thing. We have to put on pause our efforts for a clinical trial. We are ready to go. We already identify good labs that can synthesize the drugs. We are looking for investors. That's where we are right now. We are looking for people who wants to invest on these and support the clinical trial. We're talking to some potential investors, but so far we're just waiting.
Okay. Thank you. And I think for those of you who have tuned into many of our webcasts, lectures, ed days, conferences, we've tried to explain that this is a common phenomenon in the field of science where you have basic scientists discovering some really exciting work. And in order to translate that to trials is, it's known as the Valley of Death in research, and it is the work of us as families and our foundation to help our researchers be able to cross that funding gap and try to find partners, find industry, encourage families to be advocates, vote for ballot initiatives, as Dr. Muotri talked about. These are our tax dollars, and we are in control of directing where they go. And then there are also, private investors and drug companies. So we know that this isn't something that's going to necessarily be in the clinic, this year, but it gives us great hope. Even though we have to be patient with the process, there is a scientific method. It's important that it's followed for the safety of our kids.
Yep.
And we trust that you have their safety in top of mind. We have one more question in the queue, and I think that's going to take us to the top of the hour. And this comes back to a question about how microglia, how and if microglia are involved in Rett syndrome.
Yeah. So microglia, it's still controversy. There is a controversy in the field for Rett. There is an original publication suggesting that microglia might have a participation in Rett syndrome. And a couple years later, 5 other groups kind of debate if that's the case or not. All this work was done in mouse models, and mouse models have different strains, so it's unclear, who is right, who is wrong. There's a couple of evidence to support one side and then a couple of evidence to support the other side. So it's very controversial. We are trying to solve this issue by using our model here to see how important microglia would be. My feeling is that microglia might have some contribution, perhaps not as strong as astrocytes. I show you that astrocytes can actually fully revert the defects in neurons. I don't think the microglia can do that. But probably microglia can help in other ways, perhaps stabilizing or fine-tuning the circuitries. But it's very unclear, very hard to do it in the mouse model. We are applying our brain organoid model to solve this question.
Okay, thank you. And if anyone has a follow-up question, I encourage you to submit it now. Otherwise, I believe that we're going to be coming to the end of our presentation. Okay, anyone? Okay, I think we might be at the conclusion. I think you've taken us all into a great frame of mind, Dr. Muotri, about the potential, the hope, the continuity of research, some very innovative, unexplored areas. We wish you the best in your continued work. You can count on us to support you in any way that we can, and you are a real gift to our community.
Thank you so much, Paige, and thank you for the families for all the support. This is really important for us, and we'll continue to work together.
We absolutely will, and we'll invite you back with your next news and updates, and we wish you all of the best. And thank you for being with us today.
Thank you.
For our families, I just want to give a quick reminder that while we are continuing to accelerate research, and that has its own timeline, there are so many things that you can be doing with your children today to create an enriched environment and improve their quality of life. And you can continue to look to us and to our calendar for upcoming events that will give you some very practical information about ways to keep those neurons firing and wiring while we look for advances with astrocytes and glia and all of the wonderful things that our neuroscientists are working on. So please join us on May 26th for a Facebook Live on ideas for modified aquatherapy at home. This will be a very exciting program, and look at our calendar. We will continue as the summer progresses and you express your needs for the things that are coming up in your lives. We will continue to bring experts to you to help you overcome those hurdles now. And we'll continue to bring you updates from our field of clinical trials, translational research, and basic science as well. We appreciate our sponsors who committed to our foundation at the beginning of the year to provide educational programming from rettsyndrome.org. And so we do thank Acadia Pharmaceuticals, Greenwich Biosciences, and Neuron Pharmaceuticals for their support. We hope you all will turn out to join us for the virtual 5K. And look to our website for all of the news and updates. Dr. Banky and our medical advisors did post a new updated message today about some of the common questions we're hearing about care in the time of COVID. So please look for that update. It was published and updated on our website today. At any time, if you have questions or needs, you can reach out to us through our phone, website. You can email me. My email is pnuez@rettsyndrome.org, and I am always available to you, as is everybody on our team. So we all wish you well, be well, good luck with graduations, promotions, everything that is virtual, and we will use technology to continue to keep us connected in these times, and we hope that we'll be able to pick up again with our in-person ed days and family meetups soon. So in the meantime, good luck, Dr. Muotri, with everything. And this concludes our session today. Thank you.
Thanks, Paige.
Bye-bye.
Bye.